After food completes its 3 to 6 hours of churning in the stomach, the next place that it goes as in the form of chyme is to the small intestine. And remember again, it's just little bits of chyme that are emptied into the small intestine over a long period of time. And that regulation of just a little bit of the stomach contents going into the small intestine at a time is important because it gives the small intestine the necessary time to complete chemical digestion and then absorption. And we're going to see how that process works. So really quickly, the overall duties of the small intestine really center around chemical digestion. This is the place where you're going to see the largest number of different types of digestive enzymes coming into play that are going to be breaking apart chemical bonds and taking organic polymers and making organic monomers. And once we get into monomers, those are molecules that are small enough to be absorbed into the small intestine and then introduced into the blood. So not only does the small intestine really focus its efforts on chemical digestion, but it's also important because it's the principal place where absorption of those organic molecules occurs. And since there's so much chemical digestion going on, and since there's so much absorption going on, you need a lot of surface area for that to occur. And there are a number of anatomical features to the small intestine that help to increase the total amount of surface area present. One of those is the plicae circulares. And what these are basically is they're like transverse ridges, or you can think of them like circular folds on the internal lining of the small intestine. And what they do is they help to slow down the rate at which chyme moves through the small intestine. And the slower the chyme is moving through the small intestine, the longer period of time I have for absorption to occur. And that slowing down sensation not only makes the chyme move at a slower pace, but it also kind of makes it spiral through the small intestine. And that helps to increase contact of the chyme with the small intestine lining. And the more contact I have, the more opportunities I have for absorption to occur. Another a feature that really helps to increase total surface area is these finger-like projections called the villi. If I were to look at the lining of the small intestine up close, and the lumen would be out here up at the top, I would see that rather than having a lining that is just straight, I have a lining that's actually folded back and forth on itself to create these finger-like projections called villi. Villus is singular, villi is plural. And these villi obviously are there to maximize surface area. All that folding back and forth increases surface area tremendously, which means there's more places to put smooth small intestine cells that can then do absorption and digestion, etc. Now, if I look at an individual cell in one of those villi, and I were to blow that up in size, let me get this out of the way so I have a place to draw, I would see that the actual cell itself also has microvilli. It too has tiny finger-like projections on the luminal side of the cell, and again, those are there to further increase surface area. So these are our microvilli. You also will hear it called the brush border. Okay, it looks like little tiny bristles on a brush, microscopic bristles on a brush. We call it the brush border for that reason. You're going to hear me talk about the brush border quite a bit more when we get to the topic of chemical digestion in a little while. Lastly, one other thing that we have in place in the small intestine that assists with absorption are, you'll remember, these things called the lacteals. And there's a lacteal drawn for you right here. Ooh, red's not a very good color. Let's try yellow. Drawn right here for you. That lacteal, remember, is an extension of the lymphatic system, and its principal job is to assist in the absorption of lipids, to carry those lipids back up to a juncture spot with the subclavian veins and then introduce those lipids into the blood. The principal locations where all this chemical digestion and absorption is occurring varies. The small intestine is a very, very long structure. It's about roughly 2 meters long when you're alive. And it actually, after death, it increases in length to about roughly 6 to 7 meters. And that's because the smooth muscle tissue, the longitudinal smooth muscle tissue in that outer smooth muscle layer begins to relax. And as it relaxes, the small intestine lengthens out to its true length. So we got, in a living person, 2 meters long of surface area in which I can have chemical digestion and absorption occurring. And we divide out those regions of the small intestine, or we divide the small intestine out into regions, I should say, starting with the location where the small intestine meets the stomach. That upper portion is called the duodenum. And the duodenum accounts for roughly about the first 25 centimeters or so of the small intestine. So it's a relatively short area. This is the location where stomach chyme obviously enters into the small intestine. It's also where pancreatic juices are released into the small intestine and where bile enters into the small intestine. So this is the first place where we get some vigorous chemical digestion occurring, because all those pancreatic juices, remember, contain lots of digestive enzymes. The second section of the small intestine is quite a bit longer, and that's the jejunum. And we'll just call from here to here all the jejunum. And the jejunum is a region where we get further absorption occurring, and it is followed up by the last segment of the small intestine, the part of the small intestine that intersects with the large intestine, and this region of the small intestine is called the ileum. Notice how ileum is spelled I-L-E-U-M. That's different from the ileum that you learned about when you were learning about the skeletal system of the body. So we have at this junction between the ileum and the cecum, which is the first part of the large intestine, there is a sphincter or junction or valve there called the ileocecal valve. And that ileocecal valve, obviously, when it relaxes, allows any food that has not been absorbed to enter into the cecum, which is the first part of the large intestine. And at that point, we begin the process of feces formation, basically. Let's see. I think that's good enough for now, except for I will highlight that in the duodenum, again, this is the place where we get the largest amount of chemical digestion. It's also where we get the neutralization of the acidic stomach chyme. And that's done because the pancreas is dumping lots of bicarbonate ions into the duodenum, that upper region. If you're interested, the hormone that induces the release of that bicarbonate from the pancreas is secretin. Okay, so the more secretin that is secreted, the more bicarbonate is funneled from the pancreas into the small intestine. And there are cells in the duodenum that actually, when they start receiving chyme from the stomach, will begin to release secretin into the blood. That secretin is a hormone. It travels around the systemic circuit, encounters the pancreas, and encourages the pancreas to secrete more bicarbonate to neutralize the acidic chyme from the stomach. Now, once that chyme enters into the stomach, into the small intestine, not only are we having some enzymatic activities going on that we'll learn about later, but we also have motility occurring. There are some types of coordinated, rhythmic, smooth muscle contractions that are happening to at first do a lot of churning of the chyme and then later to move that whatever has not been absorbed onward towards the small intestine. And there are two distinctly different motility processes happening first. Here, the first one I'm going to talk about is segmentation. Segmentation is what you see in this first picture below here. This is the most common form of movement of the chyme within the small intestine, and the main purpose of this is to maximize contact between digestive enzymes and the chyme, and that's done by just churning it around a whole bunch. The way this works is you have segments of circular smooth muscle tissue lining or surrounding the small intestine, and two non-adjacent, meaning they're not right next to each other, two non-adjacent segments will contract at the same time. So here are two non-adjacent segments contracting at the same time, and two other non-adjacent segments next to it will relax at the same time. And then these two that had been contracting now relax, and these two that had been relaxed now contract. And we get this alternating relaxation, contraction of non-adjacent segments. And what it does is it pushes the chyme back and forth, okay, between segments, and kind of sloshes it around, and you can see that over time you get a much more uniform mixture of chyme with digestive enzymes. So this is just a sloshing and mixing motion to try to promote maximum contact between digestive enzymes and the chyme. And this can happen for quite a number of hours. Eventually, though, as we get to the point where we've more or less absorbed as much as we're going to from the food, from the chyme, at that point, then we just need to simply move that leftover food residue forward towards the large intestine. And to do that, we use peristalsis. You've already learned about peristalsis. That's where you get circular contraction behind, circular muscle relaxation in front, and the longitudinal muscles shorten up around the area of the chyme that's being moved forward. And the end result is that the chyme moves downward, just like squeezing toothpaste out of a tube. That's enough on the small intestine. We're going to move on to the large intestine and just look at some of its overall duties.